sequence is complementary, it can fold back on itself and can make something like that, where you have some base pairing even in the RNA molecule, because as it happens, the sequence in that area is complementary, because they can fold. The RNA, your RNA may be partially double-stranded, okay? So RNA is like DNA, a flexible molecule. It's not just a rod, okay, a linear or a ladder. In case of the DNA, they are flexible molecules. Now we have our genomic DNA, millions and millions of base pairs. Is all of our DNA transcribed? No, huh? What is transcribed? Only the things that we call genes. Because the genes, they contain the information that we need in form of proteins or as regulatory RNAs. Genes, they make up just a small part of our genome. The coding sequence make only a small part of our genome. A biggest part is repetitive DNA, actually. Repetitive DNA that doesn't code for anything. It's just there, and we wonder why it's there. Okay, so genes are transcribed, and actually not the full part of the gene, only the area that is important for information. What is a gene? A definition for a gene. It's not always protein. It's already too specific, because we have products that where the end product is RNA, and it's not going to the protein level. So a gene, more general, would be an information unit. And when we talk about protein, then we have one specific kind of RNA in our mind. The so-called messenger RNA or mRNA. So that is for protein coding. Genes, okay, they produce an RNA called messenger RNA. If you think about the gene, how would we recognize it? If we just have the sequence, the base sequence, how would we recognize that is a gene? What does the RNA polymerase, and we need RNA polymerases to synthesize RNA. What does the RNA polymerase need to transcribe just the gene and not some repetitive DNA? We need some regulatory sequences, like a promoter. So promoter is very important. We need a promoter that helps our RNA polymerase to bind. Normally, some other proteins help the RNA polymerase to bind there at the promoter region. Okay, so we have a promoter. Now, the transcription does not start immediately at the promoter, but there is some distance. Okay, so somewhere behind the promoter, we start transcription. So we have our transcription start site. And at some point, we also have to stop transcription. We don't want to go endlessly on and on. At some point, we have to stop the whole thing. So the termination. Termination. So we have a start of transcription at a specific site. For one specific gene, it will be always the same place where it starts and will be always the same place where it stops. Okay? So we have a start and a stop. If you compare many sequences for the point where transcription starts, you may see some kind of conservation of the base sequence, but it's not very good, let's say. Okay, it's not 100%, it's not 90%, it's not 80%, it's just somewhat conserved. Okay, so you could try to train a software to recognize, but it would not be 100% correct. You could never train your software to recognize each and every start site of all genes. Okay, and if you would switch from one organism to another, you would have to train your software again, because different kind of organism may have different preferences there. Okay, and it's also different between bacteria, where it's a bit more conserved than in eukaryote. Okay, same for termination signals. Sequence can also be quite different. Okay, so the area may be able to form some kind of stem loop structure that will block the RNA polymerase from moving on. Okay, good. Now, when we say we have messenger RNA protein coding, normally in the eukaryote, that area that is transcribed will not contain only coding sequence. It will be a mix of non-coding and coding sequence. You may have heard about intron and exon structure in the eukaryote. The bacteria very rarely have something that is looking like an intron. Very few examples, okay? So normally we say, no, they don't have it. It's not fully true. Like most often in biology, you can always find something if you look hard enough. Okay, but we say normally bacteria don't have introns, but the eukaryote have intron and exon. And intron is non-coding, okay? So that means we may have, I make that here now, something that is non-coding, an intron. Our coding area should normally start with a start codon called ATG at the DNA level, AUG at the RNA level. So that means somewhere after the transcription start, we may have a start of the coding area. At the DNA level, we have an ATG there. Actually, we can have more ATGs. Not only here at the beginning of the coding area, we can have ATGs somewhere inside, again, of the sequence, okay? So there may be several of them, but only one is used as the start codon. And somewhere we will have also a stop of the coding area, where we have a stop codon. Like, for example, the TAA or TAG or TGA. Okay. So that would mean our coding area is somewhere here, but this part here actually it has to go away because it will interrupt the coding sequence. So if we later have translation, it could not work if we leave this non-coding area there. We have to remove it by splicing. 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 That means, again, some proteins, splice factors that can recognize the beginning and the end of the intron, because there is some sequence conservation. It's also not very good, but it can be recognized by certain proteins. And they can cut the RNA on both sides of this intron sequence and remove the intron sequence and can combine the RNA again, okay, so that now the intron is gone and only the coding area now is there. What about this area here? And this area? Because we have our start of transcription here and the end of transcription here. So that means after the stop codon and in front of the start codon, we have sequences that are non-coding. They are not removed. They stay in the final transcript, in the processed transcript in our mature messenger RNA. They are called 5' and 3' untranslated regions. So that would mean that is the 5' untranslated region here. The 5' UTR untranslated region. And that one here is the 3' 3' UTR untranslated region at the RNA. Okay. They are also called leader and trailer. It's just other names for the same thing. Okay. So that's not all. What else do we need? It was found that the eukaryotic messenger RNA contains a so-called poly-A tail. Poly-A tail. That means if we have our RNA, then at the end we find a lot of A's. They're not encoded at the DNA level. They are added to the transcript by a special enzyme. A special RNA polymerase that adds the A's, that may be 100 or more A's that are added. What for? Bacteria don't have it. Eukaryotes have it for most of their transcripts. Not all. The histones, for example, are one exception. Histones have no poly-A, but most other transcripts that code for proteins, they have a poly-A. What is that good for? Has something to do with stability. Stability of the transcript. Researchers make experiment. They synthesize RNA with and without poly A and different lengths of poly A, and inject into the cell. They use the oocyte of the frog because the oocyte of the frog is quite large. So under the inverted microscope with some micromanipulator, you can inject with the glass capillary your RNA in solution a little bit. And then they looked what happened, and they found out that the RNA with the long poly A tail is stable for a longer time. So if after 1 day or 2 days or 3 days, they extract the RNA from these oocytes and look for it, they found the longer the poly A tail, the more stable the RNA is. Okay, so it has something to do with stability. Bacteria don't have stable RNA because bacteria are not interested in having stable RNA. Bacteria just want to metabolize the nutrient that is present and that gives them most energy. Okay, and so they will just produce the enzymes that they need for that. And if that nutrient is gone, they don't want to have the RNAs that code for the enzymes necessary to metabolize that around any longer. They don't want to produce that any longer. They want to save their energy. They want to be efficient. Okay, so that means their RNAs are very short-lived. They just produce the enzyme for that moment and then gone. Whereas in the eukaryote, we may have transcripts that are around for a long time. Okay. So poly A. Then another thing, this is the 5' end, and that is the 3' end. So at the 5' end, we also add something to our transcript. Yes. What is that at the 5' end that we have? It's called a cap. A cap. It's a 5' connected base that is also methylated, and that acts also as a kind of protection for our messenger RNA, and is actually also necessary to help the RNA to go out of the nucleus. Because in the eukaryote, transcription happens in the nucleus, and the RNA, the messenger RNA, has to go out of the nucleus. If you remember the structure of the nucleus, to go out of the nucleus, you have to pass the pores, the nuclear pores, and there is some kind of regulation. It's guarded, so what can go in and can go out. And without the cap, you cannot pass there. So we need it to go out of the nucleus. And it also helps us later on in translation, because it has something also to do with the orientation on the ribosomes later on. Okay. So it means our messenger RNA has to undergo several steps of processing. It has to be processed. So in the eukaryotic RNA, messenger RNA, it means we have the splicing, then we have the polyadenylation, and then we have the capping. Several steps of processing that are required to go to the mature messenger RNA. So the mature messenger RNA is normally much smaller than the first transcript. The first transcript, what they also call HN RNA, heterogeneous nuclear RNA, because it contains all the intron sequences, and introns can be very large. Introns can have more base pairs than your coding sequence. They also found cases, researchers, where another gene sits in an intron of another gene. So it can be quite complicated. Okay, let's have a look at some slides. I have to go out of that one here. So we know the gene expression, it goes from the DNA to the RNA to the protein. We have two strands of DNA. That means only one strand of the DNA will act as a template. Okay, only one strand will be the template strand. The RNA is synthesized from 5' to 3'. Okay, from 5' to 3'. The same as DNA synthesized from 5' to 3'. That is how the polymerases work. They always add to the 3' end. There is no way back. The way it goes is from DNA to RNA to protein. You may have heard that called as the central dogma, that there is no way back from the protein to the nucleic acid. Actually, it's a bad word, dogma, because we don't want to use something like that normally in science, because dogma is something that is like that and you cannot change it. It's more like religion. You have to believe that it is like that, and that's it. Don't try to discuss it. Okay, but in science, normally we want to be open. We have to be open. If new evidence comes up, we have to be able to change and adapt to the new evidence. But the central dogma is like it goes from DNA, RNA, protein, and never goes back. So far, we have never found something that allows us to go back. So the RNA polymerase needs a promoter. And it has to unwind the double-stranded DNA because it can make a complementary sequence to the template strand only when it's single-stranded. Okay? The RNA polymerase can do it by itself. It doesn't need a helicase like in replication, okay? But it needs some other proteins to bind to the promoter normally in the eukaryote. It will not bind just by itself, okay? It may be in the bacteria like that, and some viral promoter that can act where the RNA polymerase can act by itself, but in the eukaryote, you normally have some other proteins that help in the binding. Okay, so template strand, and you can see we have some initiation site, okay, where it will start. And we have a termination as well. And the termination sequence, that just show a red bar here, it may form some kind of stem loop structure that will block the RNA polymerase from going on. You may have heard about the promoter that you may have heard that they call it a Tata box, because at the beginning, the first promoters that were sequenced, it was found that they contain T and A preferentially. not all promoters have this sequence. So that is not true in general, okay? In the prokaryote, sometimes they call that Tatar sequence also a Pribnow box after the researcher who found it first. Okay, and you can see further up, they have some other sequences, what they call in the prokaryote a -35 region, and in the eukaryote CAT box, CAAT box, that is somewhat conserved, that is also required or more often found in the promoter sequence. Okay. You may see some numbering here. The numbering works that way that the transcription start site gets the +1. The first transcribed nucleotide is +1. And everything that is upstream in front of it gets negative numbers. So you can see the numbering in the promoter is minus. Okay, -10 here, -35, and so on, and the same also in the eukaryote gets negative numbers. Okay, and you call this area upstream of the transcription start site. So, and the thing that follows the transcription start site then is downstream of the transcription start site. Upstream, downstream. The eukaryotic genes may have introns. Depending on the gene, the number of introns can vary. Okay, there may be small genes that is in the previous year. So from the transcription start site downstream into then later the coding area, that thing gets positive numbers, and upstream of it into the promoter region, it gets negative numbers. So that is how it's generally done. But it's always relative to the element that you talk about. So you could also say upstream from the translation start site or downstream from the translation start site. You have to have a reference point. Okay. But the negative, that is always here for the regulatory sequences, okay? Upstream from the transcription start site. Good. So we have the splicing. Then there to get rid of the intron sequences that are in the primary RNA transcript to come to our messenger RNA. We still need the poly A and the cap. One gene, because of multiple introns and exons, may produce different transcripts because splicing can work in different ways. So the different parts of the coding region are present or absent in the transcript. Here they have for the alpha tropomyosin gene, you can see in different tissues, maybe different transcripts are produced from the same gene. They say here in striated muscle, in smooth muscle. Another one in striated muscle was found myoblast, non-muscle fibroblast, hepatoma, and brain. Always the same gene, but it may produce different transcripts, slightly different transcripts, because different exons are combined. So that means the splicing can work in different ways in different tissues from the same gene. There's nothing what bacteria can do. Only we can do that because we have this intron-exon structure. So that means we can get several different proteins from one gene. They are similar, but not identical. Okay. It's also called differential splicing. differential splicing. Okay. So that is just one.
Transcription is DNA-directed RNA synthesis,
I don't know,
catalyzed by the enzyme RNA polymerase. The DNA double helix must unwind to expose the template strand, so RNA polymerase can add the ribonucleoside triphosphate monomers to the growing RNA transcript. RNA polymerase reads the template strand from its 3' to its 5' end, and the RNA transcript grows from its own 5' to its 3' end. The DNA double helix then rewinds. When the RNA polymerase reaches the end of the template region, the enzyme and the new transcript are released.
Okay, 5' to 3'. RNA is produced. So the information, what I told you already, the information goes from DNA.
The central dogma of molecular biology, as first stated by Francis Crick, asserts that information flows from DNA to RNA, and then from RNA to protein. Information never flows from protein back up the line.
Okay, central dogma, what they call it. Until nowadays, if you happen to find some way back, then you get the Nobel Prize. Okay, sure thing. So, okay, the DNA consists of 2 base paired strands, top strand, bottom strand, and only one will act as the template strand. Okay, template strand has to have the complementary sequence to the RNA, and the RNA, as you can see, gets the U instead of the T. Okay. So an open reading frame, that is also something that you should remember. Open reading frame, our coding area, starts with an AUG, our start codon, and continues in triplets to a termination codon. In this case, they have the UAA, TAA at the DNA level. Okay. Only one reading frame is open. You have to think, if you shift by a single nucleotide and start to read 1, 2, 3, then you are in a different reading frame. You have different codons then. So only if you go from this AUG and then always take 3, 3, 3, will you be able to reach that stop codon here. If you make a shift by 1 base or 2 bases, then you go into a different reading frame and you may encounter stop codons along the way, and you will get termination like what they show here and here. Okay, so we have to be in the correct reading frame. The gene may be longer than the sequence coding for protein. What I told you, we have the non-coding sequences at the beginning, the leader, and at the end, the trailer, which are also called the 5' and 3' UTR, untranslated regions. Okay? Our RNA, which is produced in the nucleus on the DNA, has to undergo the processing, including splicing, capping, and polyadenylation, and then has to be transported out from the nucleus into the cytoplasm, where we will then have a translation happening. Okay, that is not what happens in the bacteria. Bacteria, they have no nucleus. They have no delay between transcription and translation. They can do it immediately together, more or less. Okay, but for us, we have to synthesize the full transcript in the nucleus and then transport it out into the cytoplasm to do translation. So the promoter is a control site. It doesn't contain code for protein, but it controls which kind of proteins can bind to this control site by its sequence, by its base sequence. If you mutate the sequence of the promoter, you will affect the efficiency by which proteins can bind to the promoter. You may knock out transcription. Certain mutations, certain base changes in the promoter will not allow the polymerase, the RNA polymerase, to bind there any longer. Then even you don't have any mutation in your code for the protein, you don't get the protein at all because you don't have transcription happening any longer. So you can have mutations in control sites or in the coding region. Both is possible, okay? So the protein has to bind. These control sites are also called cis sites, CIS, cis, whereas the proteins that bind there are also called trans factors, trans, T-R-A-N-S, trans factors. They come from somewhere else. The cis factor in your control site, and that is the cis factor, is in place. Okay, it's there. The proteins come from somewhere else. That's why they're sometimes also called trans factors. We are diploid organisms. We have two copies of each gene because we have one genome from the father, one genome from the mother. That's why we have 46 chromosomes. One genome is actually only 23 chromosomes. That means each gene exists in two copies. Only the one on the X and the Y chromosome, not, but the ones on the other chromosome exist in two copies. They are also called alleles. Allele, okay? So if you have the cis-acting sequence, then it affects only its own gene. The cis-acting area cannot affect the allele, the other allele, the second copy. So if you have a mutation in the promoter of one gene, it doesn't affect the second copy of the gene, okay? But if you have a mutation in the trans factor, the protein, if the gene of the protein that comes to bind to your cis-acting area in your promoter, if that was mutated, then it can affect both genes because that protein might go to both copies of the gene, okay? So it means mutations in the cis-acting area or mutations in the gene coding for the trans factors have different effects, okay? So central dogma again. The central dogma states that information nucleic acid can be perpetuated or transferred, but the transfer of information into protein is irreversible. We know we can go from RNA back to DNA by using reverse transcriptase to get a cDNA. That's what we all do very often when we code, try to clone transcripts. We make cDNA by reverse transcription, but we cannot go back from the protein. Genomes have very different sizes. Bacteria are very small, normally just a few million, but our mammals can be very large. We have some billions, 3 billion base pairs. This is an old slide. This is actually overestimated. We don't have 100,000 genes. We have only around 20,000 genes, actually. We have many genes less Researchers, before they make the genome project, they estimated that we have 100,000 or so, but it turned out that we have many genes less. But because we have the differential splicing, we can create many more proteins than we have genes, okay? So splicing removes the introns, okay, what we had already. Okay, so that should be all what we do for today, okay? Tomorrow, I will talk about regulation in bacteria, gene regulation in bacteria, and about translation process. And I had a look already at your exam result. You have to try to remember more a bit. That thing is not always good, what I had to read. Maybe it's a lot for you and you have little time. I don't know. Normally, I think what I talk to you, you should have heard that already somewhere along your life and education. That is just a repeat. But some of you have some difficulties, I think. Okay. Okay. That thing. So normally I just ask question about the thing that I also show, and I try not to be too difficult. And I have only 2 questions always for each lecture. That thing. And so I think I ask for the DNA about the experiment to demonstrate that the DNA is the nucleic acid material, the Hershey and Chase. And the other just the structure of the bDNA. And I think I talk about this, and I give you also the slide that show that that thing. Okay, so it should actually not be too difficult, okay, to answer that. So first you have to write something down. If you don't write anything down, I cannot give you any point, okay? And so you should at least try to write a bit. It doesn't need to be complete, but if you can just give some part of the answer, then I can give you some points. Okay, not 10 points, but maybe it's 5 or 6 or 7 points, something like that. That thing. But if you just write 2 words or nothing, then most likely I will give you 0 point for that. Okay, that thing. Okay, I will give you the handout from today as well. We can do that like the last time. And you didn't get that from the last lecture? The last slide from today? We were here at that slide, the last slide. Before this. Yeah, you have two genes normally because you have one copy from the father, one copy from the mother. Okay. If in one gene you have a mutation in the promoter sequence, then it will affect only that gene, because the second copy of the gene still has the normal promoter sequence. Okay, it can work normally. Transcription can work normally. But if you have a mutation in a protein that binds to the promoter in the protein, because the gene that codes for this protein carries a mutation, then this protein, because it can go to one gene or the other gene, because it's just floating around, and act as the transcription factor, let's say, in the nucleus, it may go to one or the other copy of the gene. So it can affect both. Okay? So trans factors, they may affect both copies of the allele. But if you have a mutation in the cis factor, like in the promoter region, then it affects only the single copy. That's why it's good we have two copies of a gene. The second copy may rescue us, let's say. Even we get only 50% of the protein, but it's maybe still enough enzyme activity, if it's an enzyme that we can live. Okay? Moment, I have to make here airdrops everyone. And airdrop. Okay, I have only one to airdrop to. Okay, so I just wait that all are there. And then so now I have 4. Okay, 1, 2, 3, 4. Four? Or will anyone else come? 4, then? Okay, so then I just send. So he is still waiting for Ling Ling. Who is the rabbit here? He's still waiting. This one here. Someone has to allow it, I guess. I don't know why it's still waiting. Not in this group, declined. So someone outside. Oh, okay, sorry. Okay. Okay, done. 3. Okay, I sent it 3 times. No. So don't know. I thought Ling Ling is also here in this room. That is here. Contacts only. So okay, good. Now I have to stop sharing here my screen and say goodbye. Bye bye. I will leave now.